So RP-3500 is a highly potent ATR inhibitor. So if you take a step back and think about ATR is a gene that encodes a kinase that is involved into the DNA mechanism of damage response. Basically, is a factor that protects the cells from replication stress. So if you think about removing this protection in a cell that is highly fasting replicating like cancer cells, is like inducing collapse of the replication fork. In particular, in cells that have already damage in the DNA repairing mechanisms, ATR inhibitors are particularly effective with a synthetic lethality mechanism of action. RP-3500 is a highly potent selective ATR inhibitor and entered the clinical phase back in July 2020 after we had important signals from the preclinical models. The phase 1 study was designed with a Bayesian approach, and that really allowed us to, moving from the classical 3+3 trial design, but having a very flexible schedule and be able to enroll up to more than 100 patients and really define the toxicity profile of different schedules. Early this year, the ESMO TAT conference, we presented the first comprehensive dose finding strategy for the RP-3500 single agent. We enrolled 120 patients, and we were able to explore 3 different dosing schedules. And we defined the recommended phase 2 dose for the monotherapy, which is actually 3 days on, 4 days off on a continuous schedule. And we also were able to explore 2 different schedules with a slightly reduced dose, 100 milligrams, and the same 160 milligrams, but 2 weeks on and 1 week off. Overall, the main toxicity profile that we observed was anemia, which is a classical on-target effect of this class of drugs. However, the toxicity were manageable. We observed some degree of neutropenia, but no febrile neutropenia. So we are ready with this schedule to move into the phase 2 part of the study. And also we are exploring the drug in combination with a PARP inhibitor and with chemotherapy. Interestingly, we developed these 3 dosing schedules, 2 of them to mitigate the toxicity, and we observed in all the 3 schedules a molecular response, so a drop in the ctDNA. We are expecting to present in the next coming meetings more mature activity data, which look quite exciting.